Hi everyone, welcome to the Live Longer World podcast. I am your host, Astha Jain. On this podcast, I have conversations with scientists and entrepreneurs that are transforming the field of longevity science. If you wish to stay notified of upcoming podcast releases and sign up for my newsletter, head over to livelongerworld.com. And if you want to receive extensive show notes and transcripts from each episode, you can sign up to be a premium member at livelongerworld.com/premium. My guest today is Dr. Bjorn Schumacher, whose work focuses on the central role of DNA damage in the aging process. We discuss DNA damage accumulation with age, how DNA repair mechanisms play a role, how DNA damage in the reproductive cells can tell us a lot about aging, human fertility, and reproductive capacity. We also discuss the role of p53 in cancer suppression, a new biological clock being developed in his lab, and how we should protect our skin and intestines from aging and thereby control DNA damage. A lot of us have heard the link between DNA damage and aging, and this conversation really dives into it. So I hope you enjoy my conversation with Dr. Bjorn Schumacher. Hi, Dr. Bjorn, and welcome to the Live Longer World podcast. So great to have you here today.
Thanks for having me.
So I'm very excited to talk to you about your research, which focuses a lot on DNA damage and aging. And you know, the word DNA damage and its link to aging gets thrown around a lot, but you really dive into this. And you show that, in fact, DNA damage might be playing a unifying role in all aspects of aging. And you also say that the genome is inherently unstable and constantly subject to damage. So maybe if you can talk a bit about what is the central role of DNA damage in aging?
So DNA is the blueprint of life. So every information about building and maintaining a cell, running an organism is encoded in the DNA. And it's really surprising how inherently unstable DNA is. It inflicts tens of thousands of lesions every single day. Every single cell in our body experiences about 100,000 DNA lesions every day. So it's constantly occurring. It's a fact of life that DNA is damaged and requires constant repair. And there are distinct repair mechanisms. And when they fail, people can age in an extremely accelerated fashion. So there are patients that carry mutations in single DNA repair genes, and they show the same aging-associated diseases when they're only 10 years old. And that we have when we are 80, 90 years old. Neurodegeneration, arteriosclerosis, and so on kill these patients before they reach teenage. So DNA damage is a really fundamental cause that drives the aging process. And what we increasingly learn nowadays is how many physiological processes are affected by DNA damage, by the response to DNA damage. And that's really how we learn to appreciate how complex the consequences of DNA damage are.
Fantastic. So you said something about response to DNA damage, and you also mentioned premature aging diseases, which I'll get to later. But in terms of the response to DNA damage, we do have DNA repair mechanisms. So what is happening with age? Is it that the DNA repair mechanisms are failing us, or is it that DNA damage overload is becoming too much to bear?
So that's a very interesting question. So if you imagine that there's now all the time DNA damage occurring, and repair is not perfect. It's very, very good, but nothing is perfect. So it is indeed possible that there's a gradual accumulation. And now, why is DNA repair not perfected? And that is because our somatic tissues only have to last for an individual's lifespan. And we hate to hear that, but traditionally humans are not made to live as long as we want to live nowadays, because it only mattered that we could pass on our genes to the next generation. And for humans, that until the next generation can run the business and be up and running and reproducing themselves, it takes about a human generation of like 30 years, maybe 40 years. And until then, our repair mechanisms are really good enough. In germ cells, they're of course functioning much better. So germline mutations are about 2 orders of magnitude lower than the somatic mutations in somatic cells. So yes, there are better repair mechanisms than our somatic cells have. We have them in our germ cells, but of course the proficiency of also investing into DNA repair is limited to what has been required in our evolutionary history to maintain our bodies. And other species have different DNA repair and DNA damage response mechanisms that are always adapted to what is necessary, really. And so, of course, a vision of our work is really to, in the future, being able to enhance DNA repair, augment DNA repair. But it's a very, very complex process. But we are learning how we could figure that out.
Yeah, I'm sure it's complex. I'm curious then, are you able to study if there is a point after reproductive age when DNA repair mechanisms just don't work as they used to? Is there an acceleration in, call it defects in DNA repair mechanism after reproductive age?
So it's probably a relatively slow-moving process that with time repair is not good enough to further maintain it. What we know currently is that when we look at mutation rates in humans, in human aging, mutations increase with age. They increase linearly. So that would suggest that indeed it's a gradual process that goes on through our entire lifespan. So it could be this gradual process. It's probably affected by, for example, individual aging trajectories that could play a major role there. How proficient are our repair mechanisms? But by and large, they are just sufficient for 3-4 decades. They are not sufficient for lifespans. Now we have a life expectancy of around 80 years. It looks like it's going to approach 100 years. And for that period of time, we do not have the sufficient DNA repair capacities in our human genome. And I think this is really the fundamental question, how we can augment it sufficiently to really maintain. You have to imagine that we have different requirements depending on whether we're talking about a stem cell compartment that is constantly renewing. Think of our bone marrow, of our blood regeneration, or think on the other side of the spectrum of neurons that we are born with that have to last for an entire lifespan, 100 years old with the same cell that you were born with. How can it fix all this damage? And I think this remains, because such a fundamental cause of aging, it really remains the very important target to really have therapies developed on. And there's currently really nothing that approaches aging at this very fundamental level. But this is something we are venturing into currently.
For pointing out the nuance that, different cells might require different strategies to work on DNA repair mechanisms. And I'm curious, when you've studied diseases like premature aging, one, is it that they have mutations in their DNA that's causing this damage, or is it defective DNA repair mechanisms? And also on that point that you mentioned that different cells might require different types of mechanisms, do you notice maybe defects in DNA repair in certain types of cells for some of these diseases?
Yeah, so these are very interesting questions. So there are two really distinct outcomes of DNA damage. One is mutations. Mutations are known to cause cancer because mutations, they change the gene sequence, gene function. So if you think about tumor suppressor genes or oncogenes, mutations will alter their function and cause cancer. That's why we know for many, many decades now that DNA damage is really the cause of cancer development. That's why carcinogens are invariably factors that inflict DNA damage, that lead to mutations. But then there's a really distinct consequence of DNA damage, and that is blockage of DNA metabolism. So, for example, transcription, replication can be blocked by DNA damage, and this has consequences that range from a cell being unable to generate new transcripts of a gene, so compromising its function, all the way to responses to, for example, replication fork stalling that induces cellular senescence. So then the occurrence of senescent cells in the aging body is mainly driven by this DNA damage response. Other cell types undergo apoptosis. You can have degeneration of tissues, stem cell loss. And so they are really this distinct type of outcomes. And we know that they are also inborn mutations, congenital defects in DNA repair that lead to either cancer susceptibility, so where mutation load is now greatly increased, and some patients have then a several thousand-fold induced susceptibility to develop cancer already when they are children. And on the other side, you have repair defects where you have degenerative features. Neurodegeneration is really rampant in some of these children suffering from DNA repair defects. So there are these distinct outcomes, and it distinctively affects which precise mechanism is disturbed. And here the important thing is that we need to start interfering very upstream with the problem. There's some aspect to that is avoidance of genotoxins, and another aspect is more really going into the molecular details to being able to target DNA repair processes.
Very, very interesting. You actually mentioned briefly that you're working towards something like this to be able to target different DNA repair mechanisms. I'm not sure if the work is too early or if you can speak to it.
Yeah, so we are in a very early phase at discovering mechanisms that what we have already discovered are mechanisms that enhance tolerance to DNA damage, that augment the maintenance of tissues tolerating this onslaught of DNA damage accumulation. And these are signaling mechanisms that are associated with longevity. For example, insulin-like signaling, TOR signaling plays a role there. Processes like autophagy, protein homeostasis play a major role in these responses. And here, this is the first line of interference, of interventions, where actually we can target longevity mechanisms that can really help to preserve the functionality of tissues. The first such demonstration is, for example, with calorie restriction, which also actually has a major effect on ameliorating the accelerated aging process of DNA repair defects. That is the first step. This is the really the first generation of interventions that we are aiming at. Now, the second, the future will of course be, and we are approaching that. The future will be really to mechanistically directly target the functionality of DNA repair mechanisms. That's kind of the 2.0 version of that, but we have already laid the groundwork now for the very first step of targeting longevity assurance mechanisms, DNA damage tolerance, and that is the stepping stone then to a future where we can really greatly expand the genome stability.
Very exciting. Looking forward to that. But on the point of longevity assurance mechanisms, you actually have a paper that says that delayed and accelerated aging, in fact, share longevity assurance pathways, which sounds quite confounding to people. So what are these shared pathways? And I guess what we learned from this paper?
So we coined the term of a survival response, and termed this also age to survive, because what it actually does is that now you have accumulating DNA damage with aging. And the organism responds to that. It doesn't respond by killing itself. Some individual cells do that by undergoing apoptosis, but the organism is trying to stay alive. And how do you stay alive when you have accumulation of DNA damage? What the organism does in aging is it reduces growth. It reduces endocrine signaling pathways. This is exactly the shift that you experience with age. Downregulation of a growth permissive tissue environment. And this is what these longevity assurance mechanisms do. They downregulate everything that has to do with growth. This is actually how an aging organism tries to limit the growth of tumor cells. You have because the risk of cancer directly correlates with age, because you have all these mutations accumulated. So if you had the growth permissive environment of a young organism, of a young human, in an aged person who has all these mutations accumulated, you'd have rampant cancer growth all over the place. You want to avoid that. You want to avoid that by changing the endocrine environment. And there are, of course, conflicts of that. You can extend lifespan if you reduce growth. We've learned that, for example, with dwarf mice, small mice that have a pituitary defect or that have reduced growth hormones, they actually live longer. They remain small, live longer, because they do this already very early in their lives. And with age, we do the same thing. We're trying to limit tissue growth, which comes at the expense of regeneration, because there's less capacity of stem cell compartments to regenerate. We see that in the bone marrow, for example, that increasingly become incapable of, building all the blood cells that we need, all the lineages that we need. We know that in muscle, for example, the stem cells of the muscle, they actually could still function quite well, but they don't have the endocrine environment that stimulates them. And so this is a very fine balance. Also, when you think now about therapies that would try to give you back some growth factors, because this, would help maybe some stem cell compartments, but you have inherently the risk that these mutated cells that are just waiting to receive some growth signals to become cancer cells. So it's a very, very fine balance that needs to be struck, because aging operates on so many different levels.
Right. Right. I was talking to Dr. David Gems, who spoke about the hyperfunction theory of aging, which was coined by Michael Blagasconi, which is basically talking about continuation of growth and the wild type gene that results in accelerated aging and the mTOR-driven growth. So the point you're talking about, growth and DNA damage, also just fits in perfectly there.
Right, absolutely.
On the point of DNA damage and germ cells, which are the reproductive cells, you talk about how DNA damage checkpoints functionally reside in the germline. And in fact, that every time there's a DNA damage to the germline, it triggers an innate immune response system, which triggers stress resistance, which again is a little more puzzling. So I guess I have two questions there. What then is the connection between the soma and the germline? You've touched briefly on this. And then part two is, what is this innate immune system that gets triggered? And perhaps how does that affect aging in terms of maybe delaying it?
So the germline and the soma interact very, very closely. That is true for humans as well as for very simple animals. Like we, for example, like to study the nematode C. elegans because it's very simple, but still it has the same genes, the same mechanisms, the same aging process that we have as humans. We know that the germline in humans has a major impact on the aging process. So it's known that there's communication It is unlikely that it has to do directly with fertility. It's more sophisticated than that. It's probably hormonal signals, growth factor signals, and cytokine signals that play a role there. Now we know, however, very little how this works in humans. Even in mammals, even in mice, we don't know very much because it's very, very complex. In worms, in C. elegans, this is much simpler. So we can learn principles from that. And indeed, when C. elegans, for example, carries DNA damage in the germline, then it cannot produce any offspring because germ cells very, very strictly control the integrity of the genomes that they are supposed to inherit. So they stop reproducing, and now they signal to the soma via an innate immune response that induces stress resistance in the soma. Stress resistance is always associated with longevity because a primary mechanism of how longevity assurance pathways operate is that they enhance resilience of the organism towards multiple types of stresses. And this is exactly what happens. And so now the soma can endure for longer and can expand the reproductive lifespan. So it's reproductive lifespan extension in response to impediments in the germ cells. And once the germ cells have repaired, then they can resume offspring generation during a period of their adult lives that normally reproduction has already ceased. And also vice versa. The soma surveys, actually, we found that very, very recently that the soma can survey the environment for stress factors. And these stress factors can impact the quality control of the genomes in the germ cells. And it is required to, for example, suppress, as we just showed in a current article, it suppresses the occurrence of aneuploidy. Now, in humans, we know that aneuploidy risk increases particularly with maternal age, because the control of the chromosome integrity decreases with age, even in human oocytes, right, that are formed also very early. They are already very old sometimes. They carry an increasing risk of aneuploidy. In humans, this leads to trisomy 21, for example. And the very similar processes happen in C. elegans. And we realize that this actually is controlled by somatic stress responses. And in humans, again, we know very little about. We know that there are environmental factors that impact the occurrence of aneuploidy. But we don't know how that works. And now we can learn from C. elegans how these somatic stress responses actually survey the environment and then control the integrity of the germ cells.
Wow. So what you're saying, basically, from what I understand, is that DNA damage in the germline can promote stress resistance in the soma. And even though when there's damage in the germline, there's no reproduction at that point, but when the repair mechanism kicks in, the organism can start reproducing again. At the same time, it's in fact promoted its longevity in some ways by becoming more stress resistant.
Absolutely. This is absolutely what happens. So the soma reacts to that. And there's evidence that in humans, this is rather similar. So, for example, what we know from historic records, the clearest coming from the Korean court, where it was for over many centuries, there were lifespan data recorded of the eunuchs that lived an aristocratic lifestyle, and they showed an extended lifespan by almost 20 years compared to male members of aristocratic families at the same time. And so suggesting that the very same mechanisms happen in humans. And again, the important thing, it's not about fertility, it's actually about signals, and we really should figure out what these signals are.
I see. So when you say it's not about fertility, because I remember if you think about humans, and this could be totally not related, but I guess with women and the menstrual cycle, I think sometimes when women are under stress and the menstrual cycle stops, so it sort of pushes the timeline back. I believe sometimes, and in fact, promotes boost longevity. So I'm not sure if this is the same mechanism that's going on with making the body more stress resistant.
So this is very interesting because in female reproduction, the age of menopause is correlated with lifespan. And GWAS analysis, so genome-wide association studies of very, very large numbers of women have shown that age of menopause is very much related to DNA repair gene variants. So it looks like that this is really a unifying mechanism, that DNA repair has a role in determining menopausal age, and that again has then a consequence of overall lifespan. So it is, I think, very plausible that the same germline somer interactions that we see in C. elegans are very much conserved during evolution because they are very important for reproduction. And this probably plays a major role in human aging as well. And the unifying mechanism, again, appears to be DNA repair, because human oocytes are extremely sensitive to DNA damage, which is a major issue, for example, when you think about cancer therapy. Cancer therapy mostly relies on genotoxins, on chemotherapy, radiation therapy, which inflict DNA damage. And oocytes are exquisitely sensitive to that. They undergo apoptosis, programmed cell death. And this is obviously a major problem then for these women, particularly when they are young. And I think here we really need to dig in much, much deeper in understanding the repair mechanisms, the responses in the germ cells, in the oocytes, how that interacts with the soma, how can we preserve better oocytes, how can we interfere with the signaling mechanisms between the soma and the germ cells, also the vice versa. It's very important that somatic inputs have a profound effect on oocytes that we just learned in C. elegans now. And I think we really have to make much more efforts to understand this better, and that will directly impact our understanding of human aging.
Fascinating. I can't wait to learn more as well. Very interesting. So on the point of DNA repair, you also study p53, which is a protein that plays a very crucial role in DNA repair and also tumor suppression, stopping cancer, and aging in the process. So maybe if you can talk a bit about what is p53 and what is the role it's playing in DNA repair.
So p53 is probably the gene that is researched more than any other gene in the human genome. Why is that? Because p53 is the #1 tumor suppressor that we have. About half of the human cancers overall have mutations in p53, more than in any other gene. So p53 really is the true guardian of the genome. What does p53 do? It responds to DNA damage, and it responds in a very complex way. It can induce cell cycle arrest, which is important to allow the repair mechanisms to repair before a cell starts proliferating again. It checks the quality of the cell, of the integrity. It can also induce apoptosis, programmed cell death, that is. We just talked about oocytes in humans. They are eliminated by a very closely related cousin of p53, p63, that induce apoptosis there. But in our somatic cells, it's mostly p53. It also induces cellular senescence. So the major driver of this, of the increase in cellular senescence in the aging organism. We know that heritable mutations in p53 lead to a rare disease that is characterized by an enormous susceptibility to cancer. And so p53 is really absolutely required to prevent cancer development. Now, how does that affect aging? There has been very, very interesting work that where additional copies of p53 offer additional protection from cancer. And in combinations with additional tumor suppressor genes, this can also extend lifespan. Now, this becomes important, for example, also in species that are very long-lived and very large. So think about an elephant. It's much larger than a human, many more cell divisions, and it lives really, really long. What does it do? It has many copies of p53. So it's super protected from cancer. We wish we had this type of cancer protection by just having a few more copies of p53. That would be truly amazing. We found actually that p53 is very conserved again. Also C. elegans has it already, and it teaches us what originally these mechanisms are good for. Because what p53 does in C. elegans, it controls the integrity of the germ cells. It controls whether a germ cell on the way of becoming an oocyte, for example, has properly done all the meiotic recombination that germ cells do, to bring new combinations of chromosome in the next generation, and p53 checks that. That's the most ancestral function. So it comes from the most important genome quality control that there is, which is in germ cells during oogenesis, for example. And in humans, this evolved then to really being the major, primary, and most important suppressor of cancer development.
You mentioned that p53 plays a major role in germ cells and quality control there. I guess after reproductive age then, is p53 still playing a role, or could that also be the case where linked to, I guess, aging and just p53 not playing a role with the germ cells?
There is some evidence to that in humans, variants in p53 and p63 might actually have an impact on reproductive aging. So there's some hints that this is in fact true. It's of such fundamental importance to suppress tumorigenesis that variants are generally would have very adverse effects. But it's really a baseline requirement that we have functional p53. And there are now actually, there's been a long quest of trying to therapeutically reinstate p53 function. There are very interesting developments in that because, as you can easily imagine, if there's a gene that is defective in half of all cancers, that this would be a really game changer in cancer therapy if we manage to reinstate this very fundamental suppressor of carcinogenesis, cancer development. We use, again, C. elegans because it's very conserved, all these hotspots, all these mutations you find in cancers, these residues, these hotspot residues are very conserved in C. elegans, and we're using that system to now devise strategies to actually reinstate function there, reinstate a tumor suppressor response when p53 is mutated as it is in cancer cells.
I see. So is there something that goes wrong with p53 with age? Like for people who don't have like a defect in p53, just if it's a healthy p53, is there something that's going awry with age that's maybe making the DNA repair process less effective?
So what happens in aging is that p53 might become chronically activated by a chronic DNA damage response. And chronic responses are really not particularly healthy. You see that, for example, when now you do the reverse. What happens if you hyperactivate p53? Now, you would think this is great because it suppresses tumors, and it in fact does that, but it comes at a cost, and the cost is premature aging. So a constitutive activation of p53, that really drives aging because it induces apoptosis, it induces cellular senescence. So hyperactivation can really be quite detrimental, in fact. So it really is about finding the right balance. And what I told you about having additional copies, now that's great because they're not really hyperactive. They're just much less likely to get lost and get mutated because you always have still some wild type p53 around. That's why this elephant strategy of having multiple copies works so well. It allows long lifespan and it allows a tumor-free survival. While hyperactivation, that really puts down the throttle to accelerate the aging process.
I see. That's exactly what I was going to ask you. All right, moving maybe a little bit from DNA damage and repair, you also have a new transcriptomic clock, the BIT clock, and you say that it's close to theoretical accuracy. So congrats on that. Maybe if you can tell us a bit about the clock.
Yeah, so biological clocks to assess aging are very important. Why are they important? Because biological age is very distinct from chronological age. We know that with increasing time, aging trajectories become very different. And having a biological aging clock is a prerequisite to really develop therapies that target the aging process, because the future vision is really to slow down aging, to reduce the risk for age-related diseases and extend health span. But if we want to test for that, we clearly need biomarkers of aging. And it turns out that because aging trajectories are so individualized, we cannot just have some physiological parameters because they are only a handful, and it's just not enough to really accurately, quantitatively measure the aging process. So that's why with really pioneering work from Steve Horvath, epigenetic clocks were established. So here, the epigenetic modifications on DNA, they are very manifold. There are many, many different types of different locations, different extent of epigenetic modifications. And because there are so many, you can find some that actually change consistently over the aging process. So this is the first generation now that is currently has been introduced also into the market of epigenetic clocks that can assess biological age. And that's a very important step. Now, the limitation there is that they do not allow us to understand which genes are affected by that. And that's why that was a major motivation to develop a transcriptome clock. Because here now we can really look at gene activity and which genes are contributing to the aging trajectory. And so we again use C. elegans for devising this clock because in C. elegans we know exactly the lifespan effect on hundreds of different types of interventions: nutritional intervention, genetic intervention, environmental interventions, pharmacological interventions. All these types of interventions, we know exactly how it affects the lifespan of the worm. Now, there we used for exactly those treatments all these different transcriptome gene activity measurements to at the same time look at 20,000 genes, how they change, and based on that, we defined a clock. The challenge thus far with transcriptome clocks has always been that there's a lot of variation in terms of how much transcript different animals generate or different cells generate even. And we eliminated that by binarizing the expression changes. That's why it's a binarized transcriptome clock. And there we can eliminate the inherent variance and drive down the major limitations thus far of transcriptome clocks and define a very, very accurate clock. And at the same time, we can validate that all these different interventions can be measured by this transcriptome clock. So the effect can be seen in very young adulthood. We can predict how much lifespan will be affected by an intervention, for example. We then validated that this also works for human cells, and we are very eager to now actually implement that into as a human diagnostics to really see how we can measure not only biological age, but also based on all this wealth of information that we're gathering there, to actually also really address individualized aging trajectories that in humans play a major role in, for example, being at risk for very specific age-related diseases.
Fantastic. So, is there, when you've tested these interventions in C. elegans or even humans, and tested the clocks there, have you noticed certain genes that come up that can promote that commonly promote longevity?
Yeah, so that's very interesting because with the transcriptome clock, we can actually look at the genes. And so we did exactly that, and we found that the major driver of the biological age in C. elegans, where we knew all these interventions, is the innate immune response. And that is, of course, very, very conserved in humans because in chronic inflammation is a major driver of the dysfunctionality, of the degeneration, of the damage of tissues that we see in aging. And this seems to be really a conserved, it seems to be so accurately increasing with age that this is really a conserved driver. We know that inflammatory responses are can be induced by DNA damage. So the accumulation of DNA damage that persists then can really be a driver for these chronic inflammatory responses that then really cause the damage to the tissues. And so this is the major part of this clock. The major driver genes of the biological clock are really innate immune response factors.
I see. So when you said that, the clock would be hopefully able to point out individual aging trajectories, is it pointing out, I guess, aging in different systems or how your own genes are interacting? How do you envision it being? Because I was talking to Dr. Morgan Levine, and she's working on the systems age clock, which can essentially tell you the biological age of different organs and tissues in your body. So not just the composite, but also the specific parts, so you can tell hopefully that this system is aging faster than another one, and that's also going to be individual. So how do you envision the BIT clock being?
Yeah, I think this will be very similar in terms of different tissues. They do have different aging clocks then, indeed. So that will be certainly one part of it. And what we really hope that we will be able to develop based on this is now also to see whether we find additional markers, not only of the age biomarker, but actually also associations to certain early stage disease development. So, for example, a major issue currently is that the chronic diseases of aging are diagnosed too late. So that is actually something that really prevents very early interventions that could probably be more beneficial than very late interventions. And here we would hope that because we have information on 20,000 genes at the same time, only a minor fraction of that make actually the aging clock, but we could at the same time develop then additional biomarker assays that are more reflective then of specific inflammatory responses, for example, of associations with certain disease trajectories. And that would then also allow us in intervention studies, and this I'm talking now really the future, to really know from intervention studies how they work, what are they affecting, and how much do they antagonize all aging or specific disease risks. And so I think it's extremely powerful to develop further these transcriptome clocks.
I couldn't agree with you more there. I think the more information we have on being able to not just detect diseases early, but also be able to really test what interventions are working, just the more powerful the research can be. One other paper I wanted to talk about was aging in skin. And I think that stood out to me because when I was talking to Dr. Calvin Ewald, he mentioned, damage to the extracellular matrix. And you point that out when in this paper, when it comes to skin aging, there's all this damage that's going on to the extracellular matrix. And in fact, we all know that skin is the first point of contact for DNA damage and DNA lesions to occur. So I guess maybe the question is that, have you noticed something with if you can detect skin aging that allows you to detect DNA damage in a person, or what is the connection there between skin aging and DNA damage?
So in the human skin, there is an inherent and intrinsic aging process, and there are extrinsic factors. And sunlight is the major extrinsic factor that damages our skin. Our skin is just not made really for this sunlight exposure that we're experiencing. When we look at our very close cousins in the animal kingdom, we see they have a lot more fur than we have, so they are much better protected. And we lack this protection. And so sunlight, because of this UV part of the sunlight, is really driving aging and driving mutagenesis. We have a huge problem still nowadays, although we all know how harmful UV light is. We still have UV-induced carcinogenesis in humans that to a large extent could be preventable. So there is a major impact of UV light, and it impacts both. It impacts again cancer development because of mutations, and it really promotes the aging process in the skin. So you see photos aging very clearly. You can even compare sun-exposed skin from protected skin. It will age very differently. And here, but the good side is really, there are a lot of good news from the dermatology field. One is we know about prevention, and we should really have vigorous protection from UV exposure. Then it's a very accessible organ. There are many new therapies that are coming out now. There are very encouraging results from melanoma, for example, that used to be a death sentence. Now it's largely treatable. There's also the very possibility of very early detection. So it's really everybody should take care of their skin also by having screenings. They're completely non-invasive screenings. So it's very easy to do. And the early removal of a mold, for example, can really make a huge difference. And there are also many therapies for inflammatory skin diseases that of compounds that are very complex to use for any internal organs, but they can be applied to the skin. So we have now very specific, much more improved therapies for many skin diseases. And so dermatology is a field that has really exploded with new treatments, new insights. It's a very exciting field. And so we can learn really a lot because it's such an accessible organ. It's a complex organ. It's under the influence of many environmental factors. Not only sunlight, I mean, also smoking affects the skin very gravely. And there's really a lot of new therapies that came out. So I think it's really a field to turn to.
I'll be sure to look into that and also be more vigilant about wearing my sunscreen. Last question. You have a recent paper that talks about how stress in the intestine or the gut can also change the chromosome. What is happening in the gut there? What's the paper talking about?
So that was a very exciting finding we made, that the intestine, in worms, the intestine, actually also in humans, the intestine can really sense a lot of stress and a lot of environmental factors. And so it does in worms. It's a major organ for immune responses and stress responses. And what we found is that there is a major stress response mechanism. It's a very conserved a stress response that also regulates immunity in the worm, and that is a profound impact of the quality control in germ cells. So germ cells regulate very precisely which germ cells should become oocytes to have them fertilized and which ones should be eliminated. Very similar than as in humans, oocytes are eliminated if they have the slightest a sign of damaged chromosomes. And we realize now that these intestinal stress responses, they respond to DNA damage in the germ cells, and they respond to environmental stress. So, for example, elevated temperature is a major stress factor for worms. And so these intestinal stress responses, they then are mediating the quality control of the germ cells. If you have a failed stress response in the intestine, the germ cells give rise to aneuploid progeny. Now, this in humans is equivalent to the age-dependent occurrence of trisomy 21 that is majorly influenced by maternal age, because there's an increasing risk of losing losing the accurate number of chromosomes, and the same happens under stress in worms. And so for the first time, we could show that it's really the intestinal stress responses that determine this. Now, in humans, there's a lot of circumstantial evidence about environmental factors that can impact the risk of aneuploidy, this age-dependent risk of aneuploidy. So I think that we have now the opportunity to really understand how somatic responses, stress responses in the body, control the inheritance of the accurate chromosome numbers to the next generation. And I think this will be a guide now to better understand how also in humans the age-dependent aneuploidy, for example, the occurrence of trisomy 21, can actually be controlled, and how the quality control can be improved in the aging organism.
So could it be that if you make the intestine more robust, you can increase the robustness of the quality control mechanism of the germ cells? And as a part two, have you tested stress responses in other organs and tissues as well, and whether that also has a quality control effect on the germline?
I think it will be very important to understand how these stress responses in the intestine act on the quality control in the oocytes, because they would offer a lot of therapeutic opportunities, because then you could actually use these very same mechanisms to improve quality control in the germ cells, in the oocytes. And I think because they are systemically acting and they are defined signaling mechanisms, there's a really lot of opportunity to make advances here that have therapeutic impact. Whether there are other mechanisms, so we in this case focused on the intestine, and that is very specific to the intestine, but we are greatly expanding currently this work to also look at other cell types, other tissues that contribute to the germ cell quality control. And I think there's a lot of exciting news that will be coming out in our research in the very near future.
Fantastic. I can't wait. So we've covered a lot, but any last thoughts maybe on DNA damage, germ cells that I haven't discussed, or maybe what else are you excited about when it comes to exploring DNA damage, repair mechanisms?
What is really exciting is that we have learned a lot about aging, about the genome, and about the repair mechanisms that are required for healthy aging. We've learned particularly how complex this biology is. There's no quick fix to this. DNA damage occurs constantly all the time, but we understand much, much better nowadays. We're gaining fundamentally new insight into how tissue cells repair, how they're maintained, how they interact with each other, how germ cells have such a profoundly higher capability to repair their genomes, that they're essentially immortal, that they can be passed on in humans. Since 200,000 years of modern Homo sapiens, we still are coming from the very same germ cells that our ancestors have passed on. And I think there's a lot of opportunity to get new insights. I think there's a lot of fundamental research that we are doing that is exciting, that will bring new breakthroughs. And I think we need to focus on expanding the fundamental research, the fundamental understanding of the biology of aging and the integrity of our genomes. And I think this is really where the major game changers, where the real innovations will come from, is the fundamental research that the field of aging and the field of DNA repair is currently doing.
Very excited. I can't wait either. So thank you so much for your time today. This has been fantastic. I learned a lot.
It was a great pleasure. Thanks for having me.
Hi again, everyone. I hope you enjoyed the episode and learned from it. If so, please show some support by subscribing to my YouTube channel, Live Longer World, liking the video, sharing the podcast, and leaving a review on Apple Podcasts. If you want to dive into these topics further and receive extensive show notes and transcripts from each episode, you can sign up to be a premium subscriber at livelongerworld.com/premium. To be notified of upcoming podcast releases, sign up on my website at livelongerworld.com, follow me on Twitter at livelongerworld, and Instagram at longevityfuture. Thanks for listening, stay in good health, and I'll catch you soon.